if you're talking about bone marrow, maybe you can give the next slide, Sandeep. Yes. So if you're talking about bone marrow is the organ which is situated in the inner side of the bones and fixed to the trabecula of the bones in every bone we have, in the head, spine, legs, and arms. And in the bone marrow, we have some sort of cells who are preparing some kind of bone marrow niche. These are stroma cells, and they are helping to feed all the cells and helping to protect them from damage. And in between this lies the only hematopoetic bone marrow. And if you have a look at the bone marrow of bone marrow smears under the microscope, it's a bit confusing and chaotic. Maybe have the next slide. And if you have a closer look, then we can distinguish several cells. And if you have a next slide, please, and if you have some expertise in it, you can distinguish between the cells who are going to the cell line of the granulocytes, marked with a yellow G, lymphocytes, marked with an L, erythrocytes, precursor cells of the erythrocytes, marked with an E, and there's one very big cell, which we call Megakariocyte, and that's the precursor cell of the thrombocytes and platelets. Can we have the next slide, please? This all looks very chaotic because everything happens at the same time, but it's well structured. Starting from a very interesting pluripotent stem cell, all the cells are going to a maturation. Can you check the next? Under the influence of, let's say, for example, one hormone that's called erythroprotein that is produced in the kidney and stimulating the stem cell to go to an erythrocyte. Another hormone is produced by blood, no, not blood cells, it's produced by endothelial cells of the vessels, and that is thrombopoietin. And under the influence of thrombopoietin, the stem cells develop to the direction of platelets. If we go further, some interleukins called GCSF stimulate the production of neutrophil granulocytes and others. Next step, please. Sandeep? Yeah. Other interleukins and GM-CSF is stimulating the production of macrophages and monocytes. And the last interleukins are stimulating the production of lymphocytes. So in a steady state, this is happening all at the same time, and it's very well balanced. Next slide, please. The most important cell of the bone marrow, of course, is the hematopoietic stem cell. We cannot distinguish this cell by morphologic criteria from young lymphocytes. So there is no real one-cell picture of the bone marrow who's presenting a hematopoietic stem cell. This picture shows cells which have been harvested by a PBC collection and then selected by CD34 positive selection, and then by cytotransfusion settled down to a smear and then stained. So these cells look a little damaged, and this is not the clear aspect they have in the bone marrow, but that you have an impression they look like this. And these cells are normally at about 1% of the bone marrow cells. In very young children, they have an amount which is more, 2 or 3%. And if very old adults, the amount is less. Hematopoietic stem cells have the capacity to regenerate and divide themselves. And the daughter cells, some of the daughter cells stay at the stem cell level, and other daughter cells follow the program of maturation to normal blood cells, as I showed you before. Next slide, please. As you know, erythrocytes have a limited lifetime of about 100 days. And so 1% of all the red blood cells have to be replaced every day. Erythrocytes are the cells who are taking care of the transfer of oxygen to all our organs. They are packed with HB, and you know all this. This is your everyday part of your everyday care. Next slide. Platelets are produced by the megakaryocytes. Sometimes they are excreted from the megakaryocytes. Sometimes the megakaryocyte just blow up and sets all its thrombocytes he's having in his plasma free. Thrombocytes live about only a few days. And of course, we know that already. They are part of the coagulation system, that blood is coagulating if you have a damage at your skin. Next slide. And the third cell line, which is produced in the bone marrow, are the leukocytes, which we can count in the blood, the white blood count. They are consisting of different cells. Some kind is granulocytes, some are monocytes, some eosinophil granulocytes, and some lymphocytes. Next slide. And now there's a step to the talk about immune system. The white blood count cells are those cells which are providing our immune system. We have two different systems in man, an inborn innate immune system and a learning adaptive immune system. And I tried to put you through this with some slides. Next slide. Yeah, now we have a small excursion. In the 1980s, the scientists were able to detect different epitopes on the hematopoetic cells by using monoclonal antibodies. So it's possible to define every cell with a collection of different antibodies, and so we can sort out every cell, and this is called cluster differentiation. So by the different epitopes which are presented at the membranes of the hematopoietic cells, we can sort them into different kinds. So, for example, all granulocytes have a marker which is called CD24. NK cells, and I will show you later what this is, have a marker CD16 or CD65. Monocytes have a specific marker, B-Lymphocyte and T-Lymphocytes have specific markers. And also Erythroblasts have a specific marker, and I just put it in here to show you that it's at least several hundreds of different markers who can be used to define all blood-produced cells. And some of them are only apparent during some kind of maturation, and then they disappear again. So we can, with this help, we can define not only which cell line is meant, but which part of maturation the cell is in the actual state. So next slide. Granulocytes, we have three different types. The most important are the neutrophil granulocytes. They have a very limited lifetime. They only stay 12 hours in the blood and maybe 2 days in the tissue. They are part of the innate inborn immune system. They are activated, for example, if a bacteria comes into contact with granulocytes, then they can set cytotoxic toxins free, which they carry in their granula, and they are able to phagocyte viral or bacterial antigens. They are active as soon as they got into contact with invading specimen. Next slide. Lymphocytes have a very long lifetime. Some of them live perhaps as long as man live. There are different cell types which are subsumized by the lymphocytes. One is natural killer cells. One is T lymphocyte. The name comes from the fact that they have a passage in the thymus. I will show you later. And the B lymphocytes. In former times, B lymphocytes got their name because in birds, many B lymphocytes are found in lymphoid tissue that's called bursa fabrici. So the B lymphocytes got their name from these bursa fabrici, but there is no lymphoid organ like bursa fabrici in man. So it was agreed that the names derived from bone marrow. Next slide. The first part of the lymphocyte is the NK cells. NK cells are in between innate and adaptive immune system. NK cells are ready to attack, for example, virus-infected cells. All the time, as long as they are not inhibited by cells who are expressing proteins which belong to the MHC. And the MHC, the major histocompatibility complex, is nothing else than what we call in man HLA, the human lymphocyte antigen. And if the cell is presenting an antigen which belongs to the HLA class 1, then the NK cells is stopped in his aggressivity. And so in this way, we NK cells are not attacking cells which presenting a self signal that belongs to the self. It's not we have to modify the expression that NK cells are attacking everything if they are not inhibited. There are some receptors, they are called key receptors, killer immunoglobulin receptors, who moderate the activity of key cells. Some of them are inhibiting on the NK cells, some of them are working like an activator. So it's a more complex situation with NK cells. Next slide. Monocytes are, in my eyes, the most interesting cells of the blood cells because they have very different functions and have a very broad spectrum of activity. In blood, they live about 2 days, but they are capable to invade into every tissue you can imagine. You can find them in the liver, and then these monocytes are called reticulum cells. You can find it in every lymphoid tissue, then most of them are macrophages. You can find them in working as immune cells. Then we call them either dendritic cells or antigen-presented cells. And with this activity, they are building the link to the adaptive immunity. I'll show you later how this works. And monocytes have the capability, as I told you before, to phagocyte infected cells, phagocyte bacteria, phagocyte every invading agent. And they are activated by the lipoprotein of the wall, for example, of bacteria or fungi. That's the way they are activated and work. Next slide. The third cell of the immune system are the T lymphocytes. As I told you, they have maybe have a long lifetime. T lymphocytes have that name because they're thymus derived. They are activated by the monocytes. So if in the normal state they are not active and not reacting to invading antigens, they have to be activated by the help of the monocytes, and we have very many different subtypes of the T cells. The most important are the CD8 plus T cells. These cells are cytotoxic, so they can kill, for example, virus-infected cells. They can kill fungal, and they can kill bacteria. And the other very important subgroup are the CD4 plus T cells. They are called helper cells because they have different tasks I show you later on. And there are a lot of regulatory T cells. And these regulatory T cells have the task to regulate down inflammation. So once the T cells have been activated and they're fighting against some infective agent, they cause some kind of inflammation in the body, and the inflammation and activation of the T cells have to be regulated down. So there are control cells which are regulating down the T cell function, that are regulatory T cells. Yeah. So from this slide is a bit crowded. It only shows all the cells which are produced in the bone marrow, have a passage in the blood, invade in several different tissues, lymphocytes mainly into lymphoid tissue and lymphoid organs, and other cells into liver and spleen, and all the tissue, mucosa, for example, mucosa in the gut. So they are present everywhere. And I wanted to show me the next slide, please. One really important point of T cells is that they have a passage in the thymus. The T cell production in the bone marrow has a broad variety, and we have very much different reacting T cells, and they are working on different antigens. And we have to prevent that T cells are active against self-antigens, which are, let's say, our own liver tissue or our own tissue in the kidney. So all T cells who might get active against self tissues are wiped out in the thymus. And this is done mainly before the child is born. This is an X-ray of the chest of a newborn. And can you see my file? No. On the chest X-ray, you see the shadow of the heart, but on the right side of the chest, yeah, there's a huge organ which is nearly filling half of the chest, and that's the thymus. And this reflects the importance of the function of the thymus in the newborn. The thymus is rapidly decreasing to a small organ which has the size of one lymph node, and the rest are staying the tissue thymus rest is staying behind the sternum, but in the newborn child, it's very, very important and very big, because every T cell has to pass through the thymus sometime. Okay, next slide. This slide shows how T cells are activated. The monocyte or antigen-presenting cells, now called BC, is phagocyting, let's say, an infective virus and presenting antigens of this virus in combination with parts of the HLA system, MHC1 or MHC2, and this is activating the T cells. And then there's a big change in the T cells. They are growing, they are able to multiply themselves, they produce cytotoxic toxin in the granula, and they start killing of the, they learn in this way which infective agent they have to kill. Every T cell is reacting to only one antigen. That's that's the fact in T cells, and it has to be activated by presenting some part of the self of the immune system, which is provided by the MHC or the HLA. So this shows the importance of the HLA system for the immune system. Next slide. Now we going to the B lymphocytes. B lymphocytes live for many years, maybe lifelong. They produce and increase immunoglobulins. And the activation of B cells is in cooperation with antigen-presenting cells and the T helper cells. And the T helper cells are communicating with B cells with MHC class 2 antigens. This is not HLA ABC, but DRB and DRBQ and DBP. So this, if we do an HLA typing of a patient, then you're getting results for ABC. This is class 1, and you get results for DRB and DQB and maybe DBP, not in all patients. And these are results of class 2 HLA antigens. And these class 2 antigens are those who produce the activation of B cells. Next slide. So this is the concept of activation of B cells. The CD4 positive helper cell are getting into contact with the B cells, and then the B cell is changing into a cell which is called plasma cells, and the plasma cell is producing the antibody. Each plasma cell is producing one sort of antibody which is only fixing to one antigen. So for all our immune system, we have very many different, great variety of different B cells and different T cells who face the different antigen they are fighting against. And a part of the B cells switch into so-called memory B cells. Memory B cells are not active, but they stay for a very long time in the lymphoid tissue, and as soon as there's a recontact, let's say, of the virus, the same virus, they can reactivate into plasma cells and start anew the production of immunoglobulins. So then the immune answer is much faster, and an infection may be prevented before the patient is getting sick. The antibodies, the immunoglobulins, are in our plasma in the blood, but they are present in every epithelium of the body, in the gut, in the mouth, in the nose, and even in tears of the eye, we can find antibodies produced by B cells. Next slide. So I'm coming to the conclusions. Blood immune cells have a limited lifespan, so they have to be replaced and regenerate after some time, and the regeneration is provided by the stem cells in the bone marrow. And if we do a bone marrow transplant, the key goal which we all want to arrive is to implement a donor stem cell population in the bone marrow of the patient. Humans do have two different kinds of immune system. One system we call innate, it's inborn, and it's working with out learning process. And these are granulocytes and monocytes and NK cells. They are active very early. Granulocytes are preventing us mainly for bacterial infections, monocytes and NK cells for viral and fungal infections. And the second one is we have an adaptive and learning immune system, which is provided by the T and B cells. They have to be activated by the immune cells of the innate system, but they provide a more specific and more specific aimed immune reactivity, and they can last lifelong and have a remembrance of immunity against different antigens, so that we are protected. Once they have learned the antigen, we are protected for the rest of our life again this specific virus, for example. Important is that the T and the B cells are working with the concept of self and non-self. So T cells fight against, when they are activated, against everything which is non-self, and then they might be activated, for example, by hematopoetic stem cells or hematopoetic precursor cells from a donor. They don't differentiate that they feel that these are non-self cells and they are attacking it. So if T cell from the recipient and they are taking donor cells, then it might lead to rejection of the bone marrow graft. And if it's we have T cells from the donor who have been activated by tissues of the recipient or the patient, then we see as a result, for example, GvHD. So, and every activation of the T cells is provided over the way of the HLA proteins, which are presented at the cell surface of every tissue. And the monocytes are the important link between the innate immune system and the adaptive immune system, because these are the cells who present the antigen to the T lymphocytes and the B lymphocytes to be activated. Next slide. So, yeah, there must be one more. But this is the end of my talk. And now I'm happy to take your questions. And I have a last question after your questions are answered.
Okay, now open for question. Anybody has a question, you can start. You can ask the questions.
Sir, I have one question.
Yeah.
Sir, for this, how this T-lymphocyte is activated? Can you explain me, sir?
I'm sorry, I didn't understand the question.
How this T-lymphocytes, how it gets activated?
No,
His question is how the T lymphocytes will get activated.
The B-lymphocytes. The B-lymphocytes are getting activated, but we have monocytes who have contact, let's say, with the infecting virus antigen, and the monocytes is phagocyting the virus and presenting parts of the virus to T cells. And some of the T cells are not cytotoxic T cells, but some of the T cells are T helper cells. And the T helper cells have on their surface the antigen CD4. So we can detect them as CD4 positive cells. And then we have contact of B cells and the CD4 positive T cells, which have been in contact with the monocytes who are presenting the antigen, which has to be fighted against. So in contact between monocytes and the CD4 positive T cells, the B cells is activated and producing antibodies. And B cells have the type of their antibody on the cell surface. So they the matching B cell to the antigen which is presented is getting into contact with this T cell and the monocyte, and then starts to produce this one immunoglobulin which it can produce. Did I answer your question?
Yes, sir. I got it.
Okay. So if there are no further questions, maybe we can move to my question, my last question. So my question is, why does everybody, you and me, has antibodies against the blood group which is not his self? Although he never had contact with any blood transfusions or even wrong transfusions. Do we have an idea?
Can you explain the patient again, which I didn't get the part where it's written as the non-served blood group? What does it mean by that? I'm not clear about it.
I'm sorry, I couldn't understand that. The question is, if you have a patient who has a blood group A, and you need a donor who has a blood group B, then the patient has antibodies in his blood who are anti-B. And then you have problem in infusing the bone marrow blood group B, or you even get more problems if you transfuse blood with the blood group B. But why does everybody who has a blood group A has antibodies against blood group B, although he never have been transfused and never been transfused false, so he never had contact to blood the group B. And I, in my life, for example, I have blood group A and have never been transfused the whole life, and I have antibodies against blood group B. Okay, shall I solve the question? These antibodies are not true antibodies against blood groups. They are antibodies who are produced against some bacterial surface proteins, which are cross-matching to our blood group antigens. And our immune system only allows the production of immunoglobulins who are effective against the non-self cells. The production of the product of antibodies against the self antigen, let's say in my case it would be anti-A, is not allowed by the immune system, and that's the reason in the T cell in the function of the thymus. The thymus is preventing that we are producing in normal life antibodies and immunity against the our self antigens in our body. So if we have some signs of autoimmunity, then it's reflecting the fact that our immune system is not working properly. And if you have doing bone marrow transplantation, for example, and having a mixed chimerism, then possibly you have different antigens of the HLA system, and then you have a higher risk to that antibodies against self, let's say, autohemolytic antibodies are produced. The risk is higher if you have a mixed chimerism after transplant, for example. Can you understand that?
Yes, sir. Grace had some question. Can we go for her question?
Yeah.
Grace, can you ask them? Grace, you were asking some question.